Well, one thing's for sure. If they don't get this myelin thing figured out, it's not going to be for lack of trying. Well, hello and welcome to another episode of You and Me and Multiple Sclerosis. My name is Pam. I've been living with multiple sclerosis for 39 years and then some. And as the person living with non-active secondary progressive MS, I'm still looking forward to them making some advances in remyelination, even though I'm not in the best position to take advantage of that because I have entire axons that are gone. And I think many of you are in the same boat. The first folks that would feel the benefits of remyelination therapy would be people who are fairly new to multiple sclerosis and have not undergone a lot of damage yet. But even so, you gotta crawl before you can walk, and you gotta walk before you can run. So as far as I'm concerned, the sooner we get that crawling part going, the better. And what is heartening to me is that even though nothing is ready for prime time yet, there's still been quite a bit going on. And I wanted to share with you a few articles that just came out within the last couple of months, March and May of 2024. That show the kinds of things that they're looking at right now so that you can get an idea and hopefully take some encouragement from this, that they're just not going to give up on this. It may be hard. It may not be around the corner, but they're not going to give up. So before we look at that, though, I found a good short video. It's a few years old, but I think it will still be very helpful for us since all of the articles talk about oligodendrocytes. I just thought it might be good to make sure that we all are on the same page in our understanding of what is an oligodendrocyte and why is it important, especially for people with multiple sclerosis.
Oligodendrocytes are among the last cells to form in your brain. They produce proteins that are important for strong, healthy neurons, like brain-derived neurotrophic factor, BDNF, and insulin-like growth factor 1, IGF-1. These proteins help new neurons grow and form synapses, as well as supporting existing neurons and keeping them healthy. Oligodendrocytes also have very special lipid membranes, and they use them to form the myelin sheath around neurons. This process is called myelination. Myelination isn't a fast process. At birth, there are very few brain regions with much myelination, and the process continues until you're in your 20s. Myelin was named by the German pathologist Rudolf Virchow from the Greek word myelos, meaning marrow, because when he first saw it, he thought it was actually inside the neurons. Since its discovery, we've discovered that myelin is actually wrapped around the neurons, and this makes it really useful. Why? Well, as we've discussed, neurons use electrical signals to communicate with each other. And for us to function normally, in real time, those signals have to be sent across some pretty long distances, like from the base of your spine to the tip of your toe. And it has to be done quickly. But physics puts some limitations on how quickly neurons can send their signals. The velocity of the signal depends on the diameter of the axon. The thicker the axon, the faster the signal. But we have way too many brain cells for our axons to be really thick. Our heads would be huge. So vertebrates evolved myelin to get around the problem. It keeps the ions close to the cell membrane so they can't escape, preserving the strength of the signal as it travels down the line. And there are gaps in the myelin sheath, called nodes of Ranvier, where ion channels are clustered so the signal can be maintained and amplified. This setup basically lets the electrical signal jump from node to node, so the message can move very quickly. And this is called saltatory conduction. So myelin, and therefore oligodendrocytes, are a really important part of what makes our human brain so special. And because myelin is so crucial for normal signaling in the brain and body, when something causes the myelin to break down, things can get pretty bad. Loss of myelin is part of the reason that things like strokes and spinal cord injuries can be so difficult to recover from. It's also a huge part of the disease multiple sclerosis, or MS. Researchers are trying to understand what causes MS, and the best theory we have so far is that it's actually an autoimmune disorder. Basically, our immune cells get confused, and instead of just attacking intruders, they start attacking the myelin sheath. Once the sheath gets damaged, the nerves are left exposed, and the brain can't signal properly to the muscles. This leads to all kinds of dysfunction, like weakness, numbness, and even paralysis. Scientists think that if we can figure out ways to protect or regenerate the myelin sheath, we can help stop and even reverse the symptoms of MS, which could be a huge help for people struggling with the disease in the future.
Okay, now having looked at that, let's go ahead. We'll look at the 3 papers, and I'm just going to go one right after the other so that you can see what is going on these days. Well, this one that was in Multiple Sclerosis News today is a little older than some of the others. It's from March 6, 2024, but it's reporting on this last year's ACTRIMS conference, and it says that there were 2 treatments to aid remyelination that are near clinical trials. And that's always good and promising, so let's find out more about that. CVL1001 inhibits EBP protein, while CVL2001 blocks the activity of CYP51. And if you don't know what any of that is, don't feel bad, because neither do I. But let's go ahead and see what this is all about. Convelo Therapeutics is developing 2 experimental oral treatments to promote myelin repair in multiple sclerosis, and both have shown promise in preclinical models. The company is in the final stages of safety studies in large animals. Once those studies are done, it plans to start studies that support regulatory applications to move the treatments toward clinical trials, according to Brad Lang, Convelo's Vice President of Research. And when they said large animals, I'm curious to know what they mean exactly, because I couldn't help it. I was picturing a cow, but I'm hoping that they picked something that was a little more biosimilar to humans. We'll find out, I hope. Lang discussed the development of the new treatment candidates at the America's Committee for Treatment and Research in Multiple Sclerosis, or ACTRIMS, in 2024, which was held last week, so that would have been February, virtually and in Florida, in a talk titled Identification of CVL 1001 and CVL 2001 as Selective Brain Penetrant Therapeutics for Enhanced Remyelination. In MS, inflammation in the brain and spinal cord damages the myelin sheath, a fatty covering that surrounds nerve fibers and helps them send electrical signals. Myelin damage, or demyelination, and the neurological dysfunction that results give rise to MS symptoms. There are more than 20 approved treatments for MS, but most work to reduce inflammation. No treatment has been proven to promote myelin repair or remyelination. And this, of course, is a great sadness to those of us with MS, because that's what we're all waiting for. Convelo was started in 2017 after its founders discovered most drugs with some remyelination potential were accidentally targeting enzymes involved in the molecular pathway that cells use to manufacture cholesterol, an important component of myelin. And that is actually something I wasn't aware of, that cholesterol is important to myelin, so you don't want to get your cholesterol down to 0, apparently. Promoting remyelination as a treatment for MS. Targeting these enzymes also promoted the differentiation or activation of oligodendrocytes, the brain cells chiefly responsible for making and repairing myelin. Based on the findings, researchers at Convelo have been working to find treatments to promote remyelination by blocking one of 2 proteins, CYP51 and EBP. Convelo leveraged these key findings and built a pharmacology engine and an extensive medicinal chemistry pipeline with the goal of designing safe, effective inhibitors of either CYP51 or EBP, Lang said. Adding the goal is to accelerate the differentiation of the mature oligodendrocyte and thus increase remyelination. Convelo's investigations have produced 2 new therapeutic candidates, CVL1001, which inhibits EBP, and CVL2001, which blocks the activity of CYP51. Both are orally available, with pharmacological properties that make them well suited as a once-daily drug, Lang said. Experiments in lab models have so far been promising. Research using human cells arranged in a three-dimensional structure that resembled a mini-brain showed both therapies could promote the differentiation of oligodendrocytes as designed. In a rodent model where myelin damage is triggered by injecting a toxic molecule into the spinal cord, treatment with either therapy led to significant increases in remyelination by about 50% at the most effective doses. Because this model is done by injecting a small amount of detergent that affects only a very specific region of the spinal cord, it has no real functional consequences in animals, Lang said. Now, I'm assuming the rodent model is not the large animal that they were talking about, because I've never seen mice that I would have considered large, but I'm still waiting to find out what they were talking about. But anyway, we continue. But the researchers have also tested on mice with experimental autoimmune encephalomyelitis, EAE, a lab-induced immune disease commonly used to model MS, which indicated both treatments can reduce disease severity, Lang said. We have designed novel, on-target, and safe chemical matter with strong drug-like properties, which are both efficacious in rodent and human systems, he said. And by the way, I clicked on a link that gives a summary of the talk, but there was still no mention of what the large animal was, so oh well. And here is an article that came out very recently. I'm recording this on May 10th, and this was posted on May 8th of 2024. Small molecule promotes myelin sheath repair in MS mice study. Mice saw improved motor function, nerve cell communication. A small molecule called ESI1 stimulated the repair of the myelin sheath, the fatty coating on nerve fibers that is damaged in multiple sclerosis, a study found. In an MS mouse model, this resulted in improved motor function and nerve cell communication, as well as reduced signs of disease-driving inflammation. ESI1 also enhanced myelin repair in aged, healthy mice while reversing cognitive decline. I like to hear that. That sounds very promising. Currently, there are no effective therapies to reverse myelin damage in devastating demyelinating diseases such as MS. Lead author Q. Richard Liu, PhD, scientific director at the Cincinnati Children's Hospital Medical Center's Brain Tumor Center, said in a hospital press release. These findings are significant as they offer new pathways for treatment that potentially shift the therapeutic focus from just managing symptoms to actively promoting repair and regeneration of myelin. The study, Small Molecule-Induced Epigenetic Rejuvenation Promotes SREBP condensation and overcomes barriers to CNS myelin regeneration, was published in the journal Cell. The myelin sheath is a fatty protective layer that forms around nerve fibers in the central nervous system, which comprises the brain and spinal cord. Produced by neighboring cells called oligodendrocytes, it helps to speed up the transmission of electrical impulses along nerve fibers. In MS, immune-mediated damage to the myelin sheath or demyelination impairs nerve function and communication, which gives rise to MS symptoms. So in many ways, this research seems to be strikingly similar to the one we just looked at, and yet different too. Understanding the barriers to myelin sheath repair. Evidence suggests that in areas of myelin damage, the ability of oligodendrocytes to repair the myelin sheath, remyelination, is impaired. Although defects in the growth of oligodendrocyte precursor cells were thought to be the cause, recent data indicate that mature cells also fail to properly remyelinate nerve fibers in MS lesions. The molecular barriers that prevent oligodendrocytes from producing myelin are poorly understood, and no effective treatments are available that reverse myelin damage or promote remyelination, Liu and his colleagues wrote. To better understand those barriers, the team first examined oligodendrocyte function in post-mortem tissue samples from MS patients with both active and inactive demyelinated lesions. While levels of mature oligodendrocytes in MS lesions were comparable to healthy brain regions, the cells inside lesions were epigenetically silenced. Epigenetics refers to cellular processes that control the activation or deactivation of gene expression without altering a gene's DNA sequence. Here, epigenetic silencing, or suppression of gene activity, was found to be the barrier that prevented oligodendrocytes from remyelinating nerve fibers in MS lesions. After screening a library of compounds to find those that block enzymes involved in epigenetic silencing, ES1, epigenetic silencing inhibitor 1, was identified as the most potent, boosting myelin production by about 5 times compared with the second strongest compound. When demyelination was induced in mice, ESI1 promoted remyelination and increased myelin thickness when the myelin repair processes had already begun. ESI1 also enabled the production of new myelin on regenerated nerve fibers following induced nerve damage. ESI1 was then evaluated in a well-characterized MS mouse model called experimental autoimmune encephalomyelitis, or EAE, which, like in MS patients, has immune-mediated damage to the myelin sheath. Without treatment, EAE mice developed chronic hind limb paralysis, but disease severity was markedly lower in ESI1-treated EAE mice. In the lower spinal cord, the area most affected in EAE mice, ESI1-treated animals showed more signs of remyelination and less inflammation than controls, suggesting that ESI1 may have an immunomodulatory activity, the researchers wrote. In line with these findings, ESI1 significantly improved motor function and nerve cell communication in EAE mice. ESI1 also boosted myelination in healthy aged mice, slowed age-related cognitive decline, and stimulated remyelination following demyelinating injury. To explore ESI1's potential effectiveness in MS patients, the team tested ESI1 in lab-grown brain organoids, which contain cells arranged in a three-dimensional architecture that resembles the human nervous system. Here, ESI1 promoted the activity of genes associated with myelin generation and stimulated myelin sheath extension. Mechanism of action experiments demonstrated that ESI1 triggered remyelination by blocking the activity of an enzyme called HDAC3, a known epigenetic suppressor of gene expression. But independent of HDAC3 inhibition, the compound also exerted its function by activating SREBP1A and SREBP2, proteins that drive the production of fatty acids and cholesterol in oligodendrocytes, the main components of the fatty myelin sheath. Our study highlights the potential of targeting epigenetic silencing to enable central nervous system myelin regeneration in demyelinating diseases and aging, the researchers wrote. This study is a beginning, Lou said. Prior to finding ESI1, most scientists believed that remyelination failure in MS was due to the stalled development of precursors. Now we show a proof of concept that reversing the silencing activity in oligodendrocytes present in the damaged brain can enable myelin regeneration. So this is interesting research. It looks like the scientists had to construct an artificial model of brain tissue in order to do this research, which of course makes a lot more sense than actually having to do this experiment on actual living brains. But they also used mice to see what would happen with them. So this is interesting. They're building up a lot of evidence that this is a workable new technique that might be worth putting into clinical trials. And we will have to watch and see what happens. And then finally, I saw this on May 6th, 2024. Nearly $650,000 was awarded to advance potential remyelination drug IFB-048. Therapy is designed after a blood pressure medicine to activate the ISR pathway. And they say that the National Multiple Sclerosis Society has awarded Inflectus Bioscience a grant valued at $649,601 to advance the preclinical development of IFB-048, the company's experimental treatment to promote myelin repair in multiple sclerosis. Well, let's see if we can see some similarities and differences with the two that we already looked at. This grant, along with the support from the Society, are a testimony to the potential of IFB-048 in multiple sclerosis. Pierre Mignoux, PhD, CEO of Inflectus, said in a company press release. And then we talk about, it's the same paragraph we've seen in the other ones, talking about how demyelination works. But they say that there are more than 20 approved MS treatments, but all of them reduce inflammation, and no therapy has ever been proven to actually promote the repair of damaged myelin, called remyelination. Finding new treatments that can promote remyelination is a major goal of modern MS research and the aim of this new project. Mignoux called it an opportunity to speed up the development of a promising drug candidate which could constitute a paradigm shift for the benefit of people with MS. And then they talk about the potential for myelin repair. The project, which will be led by Brian Popko, PhD, a professor at Northwestern University, will test whether IFB-048 can help promote remyelination in MS mouse models. Researchers will evaluate if IFB-048 can protect nerve fibers and/or oligodendrocytes, which are the brain cells mainly responsible for producing myelin. The experimental therapy's distribution throughout the nervous system and metabolic properties also will be evaluated. We look forward to the results of these experiments and hope that IFB-048 continues to show promise for promoting myelin repair and stopping MS progression in its tracks, said Walter Kostic, PhD, Associate Vice President of Translational Research at the National MS Society. IFB-048 is designed to activate a molecular signaling pathway called the Integrated Stress Response, or ISR. Popko said, guanabens, an approved treatment to lower blood pressure that's able to activate the ISR, has shown promising results in MS studies. IFB-048 was designed as a derivative of guanabens, the aim being to maximize the drug's ability to activate the ISR while minimizing its impact on blood pressure, which may cause unwanted side effects. In animal models of MS, we confirmed that another guanabens-related compound, IFB-088, was able to protect oligodendrocytes and myelin against the inflammatory environment, Popko said. Okay, so what we're seeing here is that many mice have given their lives to hopefully find a cure for multiple sclerosis. And so I'm just taking a moment here to thank those mice and just hope for their sake that all of this research has not been in vain. Well, finally, I have a short video clip that I thought, even though it again is a couple of years old, I liked what she had to say about the serendipity of scientific discovery. That just because you're looking at one thing doesn't mean you might not discover something else while you're at it, and that something else might turn out to be the breakthrough that we've been waiting for. So let's take a look.
So how does this actually fit in with the research on MS that Kayleen and the Menzies Institute is doing? Well, myelination, we know, is also altered in people with depression, and a lot of brain scans will show you that the white matter is altered and myelin overall in the brain is very much reduced, and there are particular areas where it gets particularly targeted. And so we thought, that's interesting, I wonder if that's happening in our mice. And so we looked at the mice that were receiving treatment with TMS And we found that in the depression model alone, without treatment, they actually had reduced myelin as well. So with that idea of myelination, we have in the mouse model of depression, we have reduced myelin. When we deliver TMS, it increases the myelin. And we have that matching with the results from Kayleen's lab showing that TMS can improve the survival of the cells that produce myelination. And this was a really unexpected finding where two completely separate research directions have come together. And it gives us a really interesting insight into how electromagnetic fields can affect the brain. They're clearly targeting myelination in some way. And also, it could be a new target for treating depression. That maybe by targeting myelination and improving that, it could also be a new way to approach depression therapeutics.
Okay, well, I think that's plenty to think about for one day, don't you? I am so excited about the future of this whole research area, and I'm going to be following it pretty carefully. And from time to time, I'm going to keep you updated on what's going on, even though, like I said, it's nothing we can run down to the doctor's office and ask for a certain pharmaceutical. There may not be a clinical trial to sign up for yet. But when that time comes, you better believe I'll be letting you know. And in the meantime, your job, as is mine, is to take really, really good care of ourselves so we are ready to take advantage of these breakthroughs when they come.